Have you ever wondered how a simple cell transforms into a mature sperm capable of creating new life? And did you know that spermiogenesis and spermiation are not the same process? These two terms often confuse biology students and even medical aspirants. In this video, we'll clearly understand what spermiogenesis is, what spermiation is, how they differ, the role of Sertoli cells, and an easy trick to remember them forever. So let's get started. First, let's understand spermatogenesis. Spermatogenesis is the complete biological process through which sperm cells are produced in the testes. This process takes place inside the highly coiled seminiferous tubules of the testes. Along the walls of these tubules are different stages of developing germ cells that gradually mature into sperm. Several hormones regulate this entire process, with follicle-stimulating hormone, FSH, luteinizing hormone, LH, and testosterone playing the most important roles. FSH stimulates the Sertoli cells, LH stimulates the Leydig cells to produce testosterone, and testosterone is essential for normal sperm production. Spermatogenesis is generally divided into four major phases. The first is the multiplication phase, where spermatogonia divide repeatedly by mitosis to increase their number. The second is the growth phase, during which spermatogonia enlarge and become primary spermatocytes. The third is the maturation phase, where primary spermatocytes undergo meiosis I and meiosis II to produce secondary spermatocytes and eventually spermatids. However, spermatids are still immature cells. They do not yet have the structure or the ability to fertilize an egg. The next stage from spermiogenesis transforms these immature spermatids into mature sperm cells. Now let's explore spermiogenesis in detail. Spermiogenesis is the process in which a spermatid undergoes a series of structural and functional changes to become a mature spermatozoon, without undergoing any further cell division. No mitosis or meiosis occurs during this stage. Instead, the cell is simply remodeled into a highly specialized reproductive cell. The first major change is the condensation of the nucleus. The nucleus becomes smaller and highly compact, protecting the genetic material while giving the sperm head a streamlined shape that improves its efficiency. The second important change is the formation of the acrosome. The acrosome develops from the Golgi apparatus and forms a cap over the head of the sperm. It contains powerful enzymes such as acrosin and hyaluronidase, which help the sperm penetrate the protective layers surrounding the egg during fertilization. Without a properly formed acrosome, successful fertilization becomes extremely difficult. Next, the tail, also known as the flagellum, develops from the centriole. The tail provides the sperm with the ability to swim through the female reproductive tract. Without a functional tail, the sperm cannot reach the egg. At the same time, numerous mitochondria arrange themselves in a spiral around the midpiece of the sperm. These mitochondria produce ATP, which supplies the energy required for continuous tail movement. Healthy mitochondria are therefore essential for sperm motility and male fertility. Another important event during spermiogenesis is the removal of excess cytoplasm. Initially, spermatids contain a large amount of cytoplasm, but as they mature, the unnecessary cytoplasm is discarded, making the sperm lighter, more streamlined, and more efficient at swimming. The discarded cytoplasm is called the residual body, which is then engulfed and removed by the Sertoli cells. Sertoli cells play a crucial role throughout sperm development. They provide nutrition, structural support, protection, and help remove waste materials from developing sperm cells. Because of these functions, Sertoli cells are often referred to as the nurse cells of the testes. At the end of spermiogenesis, a fully developed spermatozoon has been formed. However, Even though the sperm is structurally complete, it still remains attached to the Sertoli cell. This brings us to the next important process, spermiation. Spermiation is the process in which mature spermatozoa detach from the Sertoli cells and are released into the lumen of the seminiferous tubules. It is important to understand that no new sperm is formed during spermiation. The sperm has already been produced during spermiogenesis. Spermiation simply releases the mature sperm into the lumen so that it can begin its journey through the male reproductive tract. After entering the lumen, sperm travel to the epididymis, where they remain for several days. During this time, they acquire progressive motility and the ability to fertilize an egg. In other words, although sperm are structurally mature after spermiogenesis, they become functionally mature only after passing through the epididymis. Let's now compare the two processes. Spermiogenesis is the transformation of an immature spermatid into a mature sperm cell through structural modifications. Spermiation, on the other hand, is the release of that mature sperm from the Sertoli cells into the lumen of the seminiferous tubules. Spermiogenesis is about formation, while spermiation is about release. Here's an easy memory trick. Think of the word genesis, which means creation or formation. So spermiogenesis equals formation of a mature sperm. Now think of spermiation as separation or release. Spermiation equals release of the mature sperm from the Sertoli cell into the lumen. This simple trick can help you remember the difference during exams. So today, we learned what spermatogenesis is, how spermiogenesis transforms a spermatid into a mature sperm, how spermiation releases that sperm into the seminiferous tubule, the important role of Sertoli cells, and the key differences between these two processes. If you found this video helpful, don't forget to like, share, and subscribe to Zenvi Health for more easy-to-understand, science-based videos on biology, human anatomy, and health. Thank you for watching, and I'll see you in the next video.